Hello everyone, welcome to QAxon. So in this video, you will learn about action potential. Under this topic, I will first be discussing what an action potential is, followed by the phases of the action potential. And finally, we will talk about the properties of the action potential. So here we go. So what is an action potential? Now let's draw a diagram to understand this. So imagine this to be an axon, and we know that at rest the inside is negative with respect to the outside. Now let's say you apply a stimulus to this axon, a stimulus of adequate strength. What happens? There is generation of a large potential here. And this potential is called the action potential. So in other words, the action potential is the response of an excitable cell to a stimulus of adequate strength. So there is a large potential generated here, and there is a reversal of polarity, which means now the inside becomes positive with respect to the outside. And then what happens to the signal? The action potential is conducted across the axon until it reaches its destination. So in other words, even if you smell a flower or if you taste something, all these signals are not traveling in your nerves in the form of taste or smell. They're traveling as action potentials. Every stimulus is being converted into an action potential, and that is how it is being conducted. So in other words, the action potential is the code language of neurons and other excitable tissues. But the more technical definition of the action potential is this. So we define an action potential as a transient, meaning it lasts for only a short duration of time. So it is transient, it is rapid, and as we saw, it is quite large. So it's a large change in the transmembrane potential of an excitable cell, that is the RMP, with a reversal of polarity. So we saw that briefly the inside becomes positive with respect to the outside. And since it's quite large, it can be conducted across long distances. But in simple terms, the action potential is the code language of neurons and other excitable cells. So that brings us to the question, does a stimulus always produce an action potential? The answer is no. If the stimulus is not of adequate strength, then what is generated is a very small potential called a graded potential. So how is this different from an action potential? So unlike the action potential, the graded potential is small in magnitude. There is no reversal of polarity. We saw that with the action potential, there is a reversal of polarity, but that doesn't happen here. Also, this is called graded potential because its magnitude varies in relation to the strength of the stimulus. We will shortly learn that this is not the case with the action potential, whose magnitude remains constant. Now, the graded potential cannot be conducted for long distances because it is very small in magnitude. So it is essentially a local potential. There are many examples of graded potential such as receptor potential, synaptic potential, or pacemaker potential. Now, the duration of the action potential varies between different tissues. The shortest action potential is seen in nerves. For example, if you take a motor neuron, the average duration of an action potential is only 1.5 to 4 milliseconds, and this enables very fast signal transmission. On the other hand, if you look at skeletal muscle, where you don't need such rapid conduction, the action potential lasts for a slightly longer period of time. It is about 5 milliseconds. If you look at the heart, the cardiac ventricle, the action potential duration is about 200 to 400 milliseconds. Now, the action potential progresses through multiple phases. So we know that initially the cell is in a state of resting membrane potential, and then once an action potential begins, there is a phase of depolarization followed by a phase of repolarization, and eventually the cell comes back to the resting membrane potential. However, this does not happen in one or two smooth phases. It occurs in steps. So let's examine the different steps involved. So the action potential begins with initial depolarization. And then there is a phase of rapid depolarization. This is followed by the spike potential, which is the segment of the action potential above zero. Then starts the phase of repolarization, and there is a brief phase of after depolarization which falls in this phase of repolarization. And then the cell membrane goes to below the resting membrane potential, so we call that as hyperpolarization, which is followed by after hyperpolarization, and now you can see that the cell membrane has returned to the resting membrane potential. So these are the different phases of the action potential. Now let's represent these different phases of the action potential on a graph. Now this is the action potential recorded from a nerve. So the resting membrane potential is minus 70 millivolt. So this is where we begin. So this is the resting membrane potential, which is minus 70 here. Now, if you apply a stimulus of adequate strength, the nerve is going to respond with an action potential. So you can see that initially there is a small and slow rise in the membrane potential from minus 70 to minus 55 millivolt. So this slow rise is called the phase of initial depolarization. Now this value minus 55 millivolt is significant because it represents the threshold potential for this tissue. So the moment the potential reaches minus 55 millivolt, there is a spurt in depolarization. You can see that now the slope is quite steep and rapid. So this phase is called the phase of rapid depolarization. Now the portion of this graph above 0 millivolt has a special name. It's called overshoot. This portion is called overshoot or spike potential. Now the moment the potential reaches plus 35 millivolt, further depolarization does not occur. So that is the peak of the action potential, and this is constant. Now at plus 35 millivolt, you can see that there is repolarization. So there is a phase of initial rapid repolarization, which is quite steep. And then you can see that this small phase is called after depolarization. It falls within repolarization. So this is between minus 55 millivolt, the threshold, and the resting membrane potential. So that phase is called after depolarization. Now what happens is it doesn't stop there. The potential goes down further to about minus 90 millivolt. So that drop below the resting membrane potential is called hyperpolarization. And finally, it rises again to reach the resting membrane potential. So this rise is called after hyperpolarization. So these are the different phases of the action potential. Now what you should also remember is that depolarization is due to the influx of sodium ions. Sodium ions move into the cell, whereas repolarization is due to outward movement of a positive ion, which in this case is potassium. So efflux of potassium ions results in repolarization.